Hello everyone. Welcome to the third class of immunology. In this class, we are going to discuss the organs of the immune system. Now, before going forward, let us have a recap of what we learned in our previous class. So, in the previous class, we discussed about the hematopoietic stem cells, how hematopoietic stem cells are generated, where they reside, and how they self renew themselves. What are their characteristics? Then we discussed the hematopoiesis and its regulation. So how these hematopoietic stem cells they can form different type of blood cells and how these different blood cells formation is regulated at various levels. On one hand, we saw the transcription factors. They were regulating the stem cell differentiation into a number of cells. On the other hand, we also saw some environmental regulatory pathways. For example, how circadian cycle can regulate the hematopoietic activity as well as its self-renewal process. Then we discussed the lineages of the hematopoietic stem cells, the myeloid and the lymphoid lineages. We discussed the different pathways, and we also discussed the different progenitors of these both lineages. And finally, we focused on different cells, their properties and their role in immunity. So we discussed about their characteristics and their function. Now, in this third class, we will be discussing about the organs. So after the cells, we are moving one step ahead, and now we will be discussing about different organs. Okay, let's begin. So if we see the organization of the immune system, so we have two categories of organs. So one organs are known as the primary lymphoid organs, and the second category of organs are the secondary lymphoid organs. Now, what are the difference between the primary lymphoid organs and the secondary lymphoid organs? Let us check. So, primary lymphoid organs are the organs which develop the immune cells. Now, you can understand these organs as a police training institutes or as a army training colleges where our soldiers or policemen are trained. In the same manner, our immune cells they are developed in the primary lymphoid organs, and these are actually the organs where our immune cells get ready, they get training, they get environment, everything so that they can mount an appropriate immune response. Okay, so these primary lymphoid organs they provide the microenvironment. Now, what is a microenvironment? A microenvironment, you can understand it is a kind of the environment where you will provide or you will supply all sort of required growth factors, all sort of required transcription factors for the upregulation or the downregulation of the required genes, as well as the supporting cells which will guide these cells to develop into a potent immune cells. So these microenvironments they are readily available in primary lymphoid organs for hematopoietic stem cell differentiation, because hematopoietic stem cells are, as we discussed in our last class, are the mother cells which give rise to all the blood cells of the body including the RBCs, WBCs and platelets. Okay, now they primary lymphoid organs they develop all the myeloid cells, they develop RBCs, and in bone marrow they develop B lymphocytes. So myeloid cells, RBCs and B lymphocytes, all these three kind of cells are developed in bone marrow, while the T lymphocytes are developed in the thymus. So thymus is the primary lymphoid organ for T lymphocytes, while the bone marrow is the primary lymphoid organ for all myeloid cells, RBCs, and B lymphocytes. Now, if we see the secondary lymphoid organs, the secondary lymphoid organs are the site of initiation of immune response. Now, Initiation of immune response means that the organs where our most of the naive cells, they will interact with the antigen, they will get activated, and then they will produce the effector cells. So these are the site of the initiation of the immune response. Here the development is not taking place. The development of our immune cells has already been taken place inside the primary lymphoid organs. But here these organs, they provide the site of action. So basically these are the organs which you can consider them like a police stations where the criminals are dragged, they are capped, and then they are interacted with the policemen. In the same manner, in the similar manner, the antigens are exposed to naive B cells or vice versa. Okay, so these are the site of the initiation of the immune response. Once the correct antigen, the right antigen interacts with the right cell, then the activation of the immune cell happens, it starts, and then it leads to the immune response. Now this immune response is not only located within a particular secondary lymphoid organ, but it can also spread to the site of the infection, the localized as well as to the whole body as a systemic immune response. So that all depends, what kind of antigen is there. Okay, so secondary lymphoid organs, they are the site of the initiation of the immune response, while the primary lymphoid organs are the organs where our immune cells develop. Now these secondary lymphoid organs are distributed throughout the body. Okay, here in this case we have the primary lymphoid organs as a bone marrow and thymus, so they are limited, while the secondary lymphoid organs are distributed throughout the body. Now what are the different secondary lymphoid organs and how they are distributed, we will discuss today. Fine. Okay, so now let's start with the bone marrow, which is the primary lymphoid organ. So all the bones, they contain a marrow. And what is a marrow? It is the sponge-like interior of the bone, and we also call it as a medullary cavity. So if you can see, this is a long bone. In the long bone here you will see the spongy bone which contains red marrow. Okay, so you can here see the magnified view of this spongy part, and here it is the compact bone, and then you can see the sponges kind of architecture here where you will see the blood vessels. The blue one is the vein and the red one is the artery, and then here the boundary is the compact bone, which is actually the calcium rich part, okay, with the osteo blasts and the bone cells. And in this spongy area you will see the blood stem cell, which is the hematopoietic stem cells. You will see they're differentiated cells like RBCs, lymphocytes, and platelets. So everything you will see within this spongy bone. Okay, then we also have the yellow marrow. So you can see here the yellow marrow. So these marrows, they are tightly packed with the stromal cells, and these stromal cells, they are the supportive cells. They provide a support for the hematopoietic or the other stem cells to differentiate and for self renewal, as well as for the developing terminally differentiated cells. Okay, so they are the supporting cells, and you will find the hematopoietic stem cells of all the stages, right, from hematopoietic stem cells to the progenitor cells, and then the precursor cells, and finally the terminally differentiated cells. Now, the red bone marrow contains hematopoietic stem cells that can become RBCs, WBCs, or platelets. All right, so that is the red bone marrow. So you can find this red bone marrow here. And the yellow bone marrow, so you can find it here slightly at the middle. Yellow bone marrow has mostly fat, and they contain stem cells that can become cartilage, fat, or bone cells. So that is the difference between the red bone marrow and the yellow bone marrow. So all the lymphocytes, they are generated in the red bone marrow. RBCs are generated in the red bone marrow. Platelets are generated in the red bone marrow. While the fat, the bone cells and cartilages, they are generated in the yellow bone marrow. Okay. So here we are concerned about the red bone marrow because that is the site where the leukocytes are generated and then the other blood cells are generated. So our long bones, for example, femur, you can find it femur here, which is the thigh bone. Humerus, which is our arm bone. So here it is the humerus. And then we have the hip bones, ilium, right? So here it is the ilium. You can see the ilium here. You know, this part, this broad part is the ilium on both the sides. And then we have the sternum. So you can find here the sternum, which is, you know, just under your chest. So that is the, yeah, this chest bone. This is the sternum. These are the most active sites for the hematopoiesis. Okay. So hematopoietic activity in the bones, that will be there. But out of all the bones, if you compare, then comparatively higher rate of hematopoietic activity will be there in the femur, the humerus, the ilium, and the sternum. So the bone marrow is responsible for the development and replenishment of the blood cells. We already discussed this. It also maintains the pool of hematopoietic stem cells throughout the life in the vertebrates. In the last class, we discussed it in detail. And these hematopoietic stem cells, they are also found in the blood. So they are not only restricted, they are just not restricted in the bone marrow, but they are also found in the blood and probably recirculate between the bone marrow and the other tissues. So this finding has actually, given us an opportunity to isolate the hematopoietic stem cells from the peripheral blood rather than, you know, undergoing the painful process of the bone marrow aspiration. Okay. So there are several types of stromal cells. As you know, in the first slide, the stromal cells, you remember that these are the supporting cells. So there are several types of the stromal cells in the bone marrow which support the hematopoietic stem cell development. And some examples are, we have osteoblasts which generate the bones and they control the differentiation of hematopoietic stem cells. Okay, so they are not only the mother cells for the bone, but also they are also controlling the differentiation of the hematopoietic stem cells. Then we have the vascular endothelial cells, and these vascular endothelial cells, they make up the lining of the blood vessels. They also regulate the hematopoietic stem cell differentiation. We have the reticular cells. Reticular cells are the cells which form the mesh. Mesh is a kind of, reticular structure, and since these cells, they form the reticular structures, we call them as a reticular cells. And then we have the sympathetic neurons which also control the release of the hematopoietic stem cells from the bone marrow. Now I think you remember the case where in the circadian cycle, in the morning hours, there was a burst of the norepinephrine. Okay, and this norepinephrine was responsible for the egress of many differentiated cells from the bone marrow. So this is under the control of the sympathetic neurons. So here you can see the diagram where the bone is cut. It has been shown and magnified way. So here you can see this is the bone part. Okay, we have cut it and then we have shown how the different parts look like under the microscope. So this cavity is the medullary cavity. This is the medullary cavity. Okay, and in the medullary cavity you have the endosteal niche and the vascular or perivascular niche, and then you have the number of hematopoietic stem cells. Now you can see the blood vessels, one is red and one is blue. The red blood vessel is an artery and the blue blood vessel is a vein. Now if we further magnify, you will find that we have the hematopoietic stem cells which are these blue, and then we have the reticular cells. You can see they have made the reticule here. You can see the mesh like, you know, structures they have made here. And then we have the osteoblast, and here this portion, towards the periphery, we have the endosteal niche, which is the microenvironment, and then you can find some neurons, which are the sympathetic neurons. So VN is the vascular niche because they are near the blood vessels. So we sometimes call them as a perivascular, which are around the blood vessels. So this niche, the microenvironment, is the vascular environment, the niche, and here it is the endosteal niche, which is away from this vascular niche towards the periphery. So towards the center is the vascular niche, and towards the periphery is the endosteal niche. And then we have the medullary cavity. This whole cavity is the medullary cavity, and in this medullary cavity you can find the central sinus. So this is the bigger part, is the central sinus, where everything is drained here. Okay, and then it goes outside the cavity, and then you have the medullary artery, and this is the artery which supplies the blood along with the different growth factors and cells to the medullary artery. So this is how the structure is organized. Okay. Now, the endosteal niche is the lining of the bone in contact with the osteoblast. So here you can see, when we magnify it further, so this is the bone, this is the marrow, and here these are the osteoblast. This is the whole lining of the osteoblast, and on this lining, near the lining, we have this endosteal niche. So you can see from here to here we have the endosteal niche. So they are the lining the bone in contact with the osteoblast, and they are filled with progenitors of the B lymphocytes and osteoblasts. So mainly in this niche, what you will find are the B lymphocytes, which are the progenitor cells. So here you can see the progenitor cells, here you can see the osteoblast, and then you will find in this endosteal niche the perivascular niche. So here you can find the perivascular niche. So here you can see this is the perivascular niche. Okay, it is from here to here, which is around the blood vessels. So this, in the center you can see the blood vessels. Okay, and these blood vessels are lined by the endothelial cells. So these are the endothelial cells. So these endothelial cells are making a line of the blood vessels. Fine. And in between the endosteal niche and this endothelial cells, the area is known as perivascular niche. So this area is known as perivascular niche. Okay. Here you will get the quiescent and long-lived hematopoietic stem cells. And these quiescent and long-lived hematopoietic stem cells, they form majorly, they are the part majorly of the perivascular niche, right? So the endosteal niche, the perivascular niche, these two are the areas, they are the differentiated microenvironments within the bone marrow. All right. Now, progressive differentiation drives an hematopoietic stem cell from its supportive osteoblast to the more central region of the bone. So that means here you can see the hematopoietic stem cells. These hematopoietic stem cells are near the periphery. So they are in the endosteal niche. Now, as they are progressing, as they are committing towards a particular lineage, and as they are differentiating from long-term hematopoietic stem cells to short-term hematopoietic stem cells to progenitor cells and to precursor cells, slowly they are moving away from the periphery towards the center. So you can see here these arrows are showing that these hematopoietic stem cells, they are moving from the periphery towards the more central region of the bone. And more mature B cells in the central part of the bone marrow, and they exit to complete the final stages of their maturation in the spleen, because we will see further that spleen is the secondary lymphoid organ which is rich in vascular blood supply, as well as it is the first line of defense for all the blood vascular pathogens. So here the bone marrow, the B cells, they will go here, and then the final stages of maturation will complete in the spleen. While the progenitors of T cells, T cell progenitors, they exit at a very immature stage. Okay, relatively very immature stage as compared to the B cell, which go to the spleen, and they complete their development in the thymus. So you know that thymus is the primary lymphoid organ for T cells. It is the place where the T cells get mature. Okay, so they release from the bone marrow as a very immature thymocytes. Sorry, or the so thymocytes you can say, because you know they ultimately they are going to be the part of the thymus. So they are immature T cells, and then they are going and taking their residence inside the thymus, where they will complete their maturation. So fully mature myeloid and lymphoid cells, they can return to bone marrow. So once these cells, whether from the myeloid progeny or from the lymphoid progeny, once they get fully mature, they can through the circulation return to the bone marrow. Plasma cells, which are the effector cells of the B cells, and which release the secretory form of antibodies, they may even take up long term residence in the bone marrow. So, once the plasma cells are formed, that means the mother B cells has already encountered an antigen, and then it has differentiated into the effector plasma cell. Now the plasma cell can secrete the antibodies, or it is secreting the antibodies, so it can also come to the bone marrow, and then it can take up long term residence, so that if any infection comes in the bone marrow, they can easily deal with it. Okay, with age, fat cells occupy around more than 50% of the bone marrow. So as we age and as we get old, our bone marrow cells, they are slowly being replaced by the fat cells, and more than 50% of the bone marrow can have the fat cells. So the efficiency of the hematopoiesis decreases. So now we don't have, the much healing power or the much regenerative power of the blood cells remain as we age. Okay, the more we age, the more efficiency goes down. Okay, so now focus on the thymus. So thymus is the the primary lymphoid organ for the production of T cells. Okay, this is the organ where our T cells are developed. And in the picture you can see here we have the anatomical sites. Here we have the trachea, esophagus, ribs which are cut. Then there is a right lung, left lung, then this is heart, and this is thymus. So thymus is the place which is the site of T cell development, and it is located just above the heart. So maturation site, and it is the largest prior to puberty. That means before adolescent, the size of the thymus is largest, but as the onset of the adolescence occurs, then the size gradually reduces. The T cell precursors, which are the precursors, not the mature T cells. So the precursor T cells, which are still pluripotent, they travel from the bone marrow to the thymus via blood, and it provides a specialized environment where immature T cells, which are known as thymocytes in the thymus and which make up the majority of the cells, they generate unique T cell receptors. And these T cell receptors are as unique as good as the B cell receptors, which bind to the M.Sc presented peptides. So they get maturity, they get the microenvironment which helps in the maturation of the T cells. And then they are also tested for their reactivity to self MHC peptide complexes on thymic stromal cells. So thymic stromal cells are the supportive cells, and they test whether the TCRs generated on the T cells, they are self reactive or not. If they are self reactive, then it may create a problem. They may produce the immune dysfunction. But if they are not self reactive, Okay, then they are allowed for further maturation. So this is known as quality check, and it it is also kind of the selective process, the selection process. So thymocytes with TCR binding self MHC peptide complexes with either too high or too low affinity, they are induced to die, and this selection is known as negative selection, because here the cells which are either very strongly reactive or which are very feebly reactive, they are not allowed to live, and they are induced to die. So that's why this selection is known as negative selection. Whereas if the TCRs binding self MHC peptide complexes with intermediate affinity are found, then these thymocytes are permitted to survive, and this is known as the positive selection. Okay, so only thymocytes which have the intermediate affinity towards the self MHC peptide complexes, they are allowed to live, whereas the TCRs or the T cells which have very low or very high affinity towards the self MHC peptide complexes, they are allowed to die. So this positive and negative selection helps to narrow down the cells which will be in our circulation. So they proceed to maturation and migration to thymic medulla. So thymic medulla, it's a part of thymus where they finally go, and then they migrate. Okay. Now, if we see, so the fresh T cell precursors in the thymus exhibit no expression of CD4 or CD8 markers. Now, I'm sure in our previous discussion, we discussed the CD4 and CD8 unique markers of the T cells, whereas the CD4 markers were attributed to T helper cells, while the CD8 markers were attributed to T cytotoxic cells or TC cells. So, the fresh T cell precursors, these T cell precursors, since they don't have any expression of either of these two markers, these cells are known as double negative. They are known as double negative cells. So we call them as a DN cells. Now when DN cells, they move to subcapsular cortex. So if you see here, this is the capsule. Okay. So this portion is the capsule. And this part is the subcellular cortex. So this is the part of the cortex, but since it is under the capsule, we call it as a subcapsular cortex. And in this subcapsular cortex part, which is from here to here, you can see here, the cells are the DN cells, and these DN cells are the double negative cells. Okay, so these double negative cells, they are found here in the subcapsular cortex. They proliferate and they move to cortex and generate TCRs. So now DN cells, they are moving to the cortex. This is the cortical part, and here in the cortex they are generating their TCRs. Okay, and slowly they are under the progressive development stage. So initially cells express both CD4 and CD8. So now they have both the markers, CD4 as well as CD8 on the single cell. So they are called double positive cells, DP cells. So here you can see in the cortical part, this is the DP cell. This is the double positive cell. Okay, because this cell is expressing both the CD8 as well as the CD4 markers. Fine. The cortical thymic epithelial cell, we call it as a CTEX, cortical thymic epithelial cells. They have long processes just like the dendritic cells, and they are read by thymocytes to test TCRs, MHC peptide complex binding. So here is the stage where the cortical means the cells belonging to the cortex region, thymic epithelial cells, these are the name of the cells. These cells, they test whether these thymocytes, because now they have both CD4 as well as CD8 plus TCRs, whether they can either bind with the self peptide MHC complex with low affinity, high affinity, or intermediate affinity. If they are binding with the intermediate affinity, they are allowed to live. But if they are binding with the low and high affinity, then they will be dead. So positive and negative selection happens here. After passing selection, cells move to medulla to meet another stromal cells, and these cells are known as medullary thymic epithelial cells. So if these thymic epithelial cells belong to cortex, we call them cortical thymic epithelial cells. But in medulla, these thymic epithelial cells are known as medullary thymic epithelial cells. And these medullary thymic epithelial cells, they support the final T cell maturation. Okay, remember that cortical thymic epithelial cells, they are actually selecting the cells by positive and negative selection, while the medullary thymic epithelial cells, they are supporting the final T cell maturation, and here thymocytes are tested for the reactivity to tissue specific antigens. So here the final selection is taking place with the help of the double negative, the double positive, and whether the tissue specific antigens, because various tissues specific antigens are brought here, and then they are selected on the basis of whether they are reacting or they are not reacting. So this is the final phase of their maturation. So negative selection can happen in any of the microenvironments. Now this is important because either they are in the cortical region or they are in the medullary region. Negative selection can happen because based on whether the affinity is low or affinity is high. Fine. Final maturation in periphery when exposed to antigens presented in secondary lymphoid organs. So now when they have qualified the selection test, then the final maturation happens in the periphery when exposed to antigens presented in secondary lymphoid organs, and most of these antigens are the tissue specific antigens. So mature cells now express only either CD4 or CD8, and we call them as a single positive cells. They leave the thymus via corticomedullary junction. So this is the corticomedullary junction, and the cells you can see is the SP cells. These SP cells, they are either the CD4 positive cells or they are the CD8 positive cells. So we call them as a single positive cells, and that's how the maturation of the thymocytes take place. Now there is a discovery of secondary or second thymus also in mice, and this was discovered in 2006. So this was a new anatomical discovery. Okay, this is a new site in the neck near the cervical vertebra of mice. Okay, near the cervical vertebra of mice. It consists of a single lobe or a cluster of single lobes and is populated by relatively more mature thymocytes. So if you remember the first thymus, it contains immature thymocytes which had some sort of pluripotency. But here in this case, the second cervical thymus, it has relatively more mature thymocytes. So that is the difference. Then it consists of a single lobe or a cluster of single lobes. Okay, so here it is a monolobular structure. Cervical thymus develops after birth and grow to the size of a small lymph node. So it is just a very small lymph node, you know, size of structure, and it develops after birth. Important is it develops after birth, and it has a typical medulla cortex structure because our thymus also has the cortex and the medulla structure, so it also has a medullary cortex structure, and it also supports the T cell development. So their role is also to support T cell development. It is also found, apart from the mice, also found in koala and kangaroo. So here you can see the picture of the koala and kangaroo, and both are found uh, majorly in the Australian region. Okay. Now, since this is the second thymus, then if you remove the first thymus, then the mice we generally called is as a athymic mice. Why? Because the mice has no thymus, and that compromises the immune response capability. But this is not 100% compromised. The reason is the mice has a second thymus, and this second thymus somehow maintains some T cells which can be helpful during the infection process. Now we are moving to the secondary lymphoid organs. So in the primary lymphoid organs, we covered the bone marrow and the thymus. Now we are moving to the secondary lymphoid organs. So these are the site of the initiation of the immune response, right? They were the sites of the development of the immune cells. They are distributed throughout the body. You take any part of the body, you will find the secondary lymphoid organs, and we have two categories. One is the highly organized lymph node and spleen. So here you can see the whole lymphatic system, and these small dots are the lymph node, and they are compartmentalized by fibrous capsules. So both the lymph nodes as well as the spleen, they have the fibrous capsule. So that is the characteristic. They have the fibrous capsule, and this fibrous capsule makes many compartments inside the structure. So in the lymph node also you will find compartments, and in the spleen also you will find compartments. While the less organized secondary lymphoid organs are known as MALT, which are the mucosa associated lymphoid tissue. MALT. And these, some of the examples are tonsils, Peyer's patches, which are present in the small intestine, and then appendix, apart from some other which we will discuss. So all secondary lymphoid organs, they include distinct regions of T cells and B cell activity. Now it is important to note here that secondary lymphoid organs, they are the site of initiation of immune response, right? So if they are the site of initiation of immune response, that means that they have to deal with the T cells and the B cells. They are the major soldiers here, and these naive T cells and naive B cells, they will interact with the antigens which will be brought here, or they will come naturally, right? So all the secondary lymphoid organs, they have the distinct regions of T cells and B cells activity. There are certain regions which will be rich in T cells. There are certain regions in the SLOs or secondary lymphoid organs which are rich in B cells, and they both exhibit the very specific T cell and B cell activities here. All have lymphoid follicle. Now you should understand this term because we will be using this particular term in our subsequent discussions. So lymphoid follicles are what? These are the highly organized microenvironments for the development and selection of B cells. Okay, so you remember that whenever we are talking about the lymphoid organs, we are talking about very specific microenvironments within the secondary lymphoid organs, where you will find a number of B cells developing there as well as selecting for a particular antigen, and these are known as lymphoid follicles. Okay. Now, if we see the blood and lymph connect, so the blood and lymphatic systems, they connect all lymphoid organs with each other and with each tissue. That means our lymphoid organs or our lymphatic system, where which has a flow of the lymph, and 99% of the leukocytes or you will be finding there. So we have these two connections, the blood vascular system and the lymphatic systems. They don't work in solo, they work in coordination, and they have a connect where the lymph can be flown from the lymphatic system and can merge with the blood, and from the blood the plasma can come to the lymphatic vessels. So the immune cells are the most mobile cells. They use both systems to move. They navigate through both these systems, the blood vascular system as well as the lymphatic system. The largest lymphatic vessel, the largest lymphatic vessel is the thoracic duct. Okay, remember, the largest lymphatic vessel is the thoracic duct. It collects the lymph from all the body except the right arm and right side of the head. So lymphatic collection from the all the body except the right arm and right side of the head is done by thoracic duct, which is the largest lymphatic vessel. So here you can see in the diagram, this is the thoracic duct, the red color one. This is the thoracic duct, and it empties into the left subclavian vein. So here is the left subclavian vein. So this is going and it is emptying into this vein here in the left subclavian vein. Okay. Lymph from right arm and right side of the head is collected into the right lymphatic duct. So we have here, you can see the right lymphatic duct. These are the right lymphatic duct which drains into the right subclavian vein, and this is the right subclavian vein. So this right lymphatic duct, it empties its content into the right subclavian vein. So remember that thoracic duct, it empties into left subclavian vein, and the right lymphatic duct, it empties into the right subclavian vein. All cells and fluid from the lymph are returned into the blood. So it keeps circulating. The lymph and the blood, they are so connected that all the cells and all the fluid, it is mixed with the blood, and it keeps, and it is drained into the blood. Steady state levels of fluids within the circulatory system. So they maintain a kind of a steady state. Steady state means they maintain a equilibrium situation. Okay. Now, if we compare the blood vascular system and lymphatic system, we will find that the blood vascular system has the networks of arteries, veins, and capillaries, and pumping is done by heart in blood vascular system. While in case of lymphatic system, it is a network of thin walled vessels which are filled with a protein rich fluid. We call them as a lymph. This protein rich fluid is called lymph, and it is derived from the blood plasma. There is no heart. Okay, there is no heart in the lymphatic system, but the flow is achieved by the movement of the surrounding muscles. So therefore we should exercise more. The more we exercise, the more movement of this lymphatic system happens, and the more healthy immune system can be achieved inside the body. And that's why the doctors say that we should go for the exercise, right? Now, blood vascular system has access to every organ and tissue. Each and every organ and each and every tissue is innervated or has access of the blood vascular system. While in case of lymphatic system, plasma, which is the part of the blood, acellular part, most of it returns to the blood, some into primary vessels, okay, and this is known as lymph, which is a large, which goes into as a large lymphatic vessels inside that. And in the lymphatic vessels, we have the one-way valves, which maintains the unidirectional flow of the lymph. Okay. Blood vascular system is lined by endothelial cells, which are very, very responsive to inflammatory signals because they have the receptors, particularly for the complements and some other cytokines which causes inflammation. So they are very responsive, and that's the reason you must have seen that wherever there is an inflammation, you will find redness there. So redness is always associated with inflammation. The reason is our our endothelial cells, they are highly responsive to the inflammatory molecules, and as soon as the inflammatory molecules they build up at the site of the infection, they bind to the endothelial cells, and the endothelial cells increases the blood flow. It leads to the heat of the localized heat. It leads to the redness. Okay, so that is because of this responsive nature of the endothelial cells. While here the primary vessels, in case of the lymphatic system, they are like blood capillaries, and you know that capillaries are very thin. So the primary vessels are very thin, and there is a single layer of loosely opposed endothelial cells, so very loosely opposed, and they are porous. Okay, so that the cells and the plasma can easily migrate. Now, in case of blood vascular system, most lymphocytes enter the secondary lymphoid organs via specialized blood vessels. So, for the secondary lymphoid organs, we have specialized blood vessels, and the lymphocytes, they enter into secondary lymphoid organs through them. While in case of lymphatic system, most lymphocytes, they leave secondary lymphoid organs via lymphatic system. So via lymphatic system, they again re-enter into the blood vascular system, and from blood vascular system, through the special blood vessels, they enter into the secondary lymphoid organs. So this is just a comparison between the blood vascular system and the lymphatic system. Now, only WBCs have access to the lymphatic system. Okay, so in the lymphatic system, you will not find the RBCs, or by mistake, or some RBCs are there, but they will be very less. Okay, but this is lymphatic system is completely, dominated by the lymphocytes. So here you can see, this is the lymphatic vessels, and these lymphatic vessels have the lymph nodes after some certain, distances. So they are just like, thread and beads, thread and beads, thread and beads like structure. So this is a reticular structure or highly networked cells, where you have, these lymph nodes. They are all around your body, and these lymph nodes are the secondary lymphoid organs and are very organized system, and they are the 100% dedicated for immune response. Now, if you see here, just how these are connected. So this is the arteriole, and then these are the blood capillaries, and here it is the venule, and you can see we also have the lymphatic system here. And so we have the lymph here, we have the blood here, we have the veins, the arteries, and then we have this interstitial fluid within the tissue. So this the interstitial fluid is, it is present in the tissue, or the tissue fluid. So we have the well organized system here. Okay, so most secondary lymphoid tissues are situated along the vessels of the lymphatic system. So this you can see here, these are the lymph nodes, and these lymph nodes, they are situated along the lymphatic vessels in the system. There are exception. An exception is the spleen. Spleen is, although it is the secondary lymphoid organ, and it is a highly organized secondary lymphoid organ, but it's served primarily by blood vessels. It's served primarily by blood vessels, not by the lymph. Okay, and why it is so, I will tell you. The lymphatic capillaries, they pick up interstitial fluid. So this is the interstitial fluid. These interstitial fluid can enter the lymphatic system. Particulate proteins. So particulate proteins can be an antigen, a particle, or a part, or the bacteria or virus. Soluble proteins, some proteins which are soluble, they are maybe antigenic peptides. They are also picked up by lymphatic capillaries, and immune cells from the tissue surrounding the blood capillaries. So the lymphatic capillaries, they pick up all these things: interstitial fluid, particulate proteins, soluble proteins, and immune cells. Now, this is the general scheme of immune response in the secondary lymphoid organ, how it happens. Now, you can see here we have foreign antigen. Okay. Now, this foreign antigen can enter tissue either independently or it can be taken up by the antigen presenting cell. So an antigen presenting cell, it will engulf the antigen. It will also take it to the tissue. So ultimately the entry of antigen into the tissue is of prime importance. Once the antigen is inside the tissue, then it goes through the lymphatic system and reaches either the lymph node or any other secondary lymphoid organ. Here in the lymph node or any secondary lymphoid organ, we have antigen specific lymphocytes, we have dendritic cells, and we have the macrophages. Now these antigen, it gets there is an interaction between the antigen and these any of the or all of these cells, and then these cells are activated, and once they are activated they give the immune response. So this is a very general scheme of immune response that happens inside any secondary lymphoid organ. Now let's talk about the lymph nodes. So lymph nodes are the most specialized secondary lymphoid organs. They are the most specialized secondary lymphoid organs. Remember, their only function is to provide protection, providing microenvironment and regulating the immune response. That is the only important function and the only function of that. So here you can see it is a network of lymphatic tissue and lymph nodes here. So we have the lymph vessels here, green color thread like vessels, and on dot 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 or these the nodular structures, these are the lymph nodes. So you can see how lymph nodes are scattered all around the body. Now if we see slightly a magnified view, so we will find that these lymph nodes are just like a small beads, and these beads are placed on these lymphatic vessels here. Okay, so these are the first organized lymphoid structure to encounter antigens that enter the tissue space. In any tissue, whenever there is an infection, that will definitely come into the respective lymph node or the localized lymph node of that region. That's why you must have heard that children usually develop tonsils. Now tonsils are what? These are also the secondary lymphoid organs. Okay, and these tonsils, they doctors say the patient is suffering with tonsillitis. So litis means whenever we have a suffix itis, that means there is an inflammation. So inflammation of the tonsils, like we have conjunctivitis. If we have inflammation in the conjunctiva of the eye, then we call it as a conjunctivitis. The suffix is itis. In the same manner, we have tonsillitis or we have hepatitis. Okay, inflammation in the liver. Okay, nephritis, inflammation in the kidney. So these are the terms. Bronchitis, inflammation in the our bronchi. Okay, so inflammation is what? Inflammation is the indication of the infection. So that means there is an infection, localized infection in the throat, and the tonsils are inflamed because there is a very high intensity immune responses going on. So the lymph nodes, wherever they are the first structures to encounter antigens that enter the tissue space. These are encapsulated. Okay, these are encapsulated. These are bean shaped structures, and they have a network of stromal cells. They have lymphocytes, macrophages, and they have the dendritic cells. So these structures, they have the networks of stromal cells, and the stromal cells are the supportive structures. These are the supportive cells, apart from the immune cells, which are the lymphocytes, macrophages, and dendritic cells. These are connected to both blood vessels and the lymphatic vessels. Connected to both blood vessels and lymphatic vessels. They are divided into three roughly concentric regions. So if we see the structure of the lymph node, any lymph node has three roughly concentric regions. Concentric means the center is the same. So this is the center. So this is one region. This is another region. Its center is also this same. And then it is a third region. This is also the same. So we have the center. Then around the center one region, around the same center another region, around the same center we have the third region. So we call it as a concentric regions. And these concentric regions are the cortex, the paracortex and the medulla. So here you will find the cortex. This is the cortical part. Okay, this is sky blue part. This cortex, this is cortex, cortex, cortex. Then you have the paracortex. So paracortex here, you can find it here. You know, the area around the cortex is the paracortex. So this is the yellow part. And then you have the medulla. This is the medulla. This is the interior part. Okay. So we have the cortex, we have the paracortex, and then we have the medulla. Now, just to elaborate the structure, you can see this is the capsule, okay, and then this is the subcapsular sinus. Sinus means space. So this is the subcellular sinus. Then we have the incoming lymphatics, which is the afferent, okay, and then we have the outgoing lymphatics, which is the efferent. So this is the afferent vessel. Afferent means which is incoming, it is coming inside, it is supplying in the lymph node. And efferent means it is outgoing, it is taking the lymph out from the lymph node. Then we have the antigen or the APC, they are coming from the incoming lymphatics. Okay, here you can see this is the blood endothelial cells. These are the organized in the blood vessels. We have the blood endothelial cells. So this red one is the blood vessel. Okay, so you can see the artery, because red is denoting the artery, and artery is the supplying blood vessel. So native lymphocytes, they are entering into the lymph node through this supplying artery, and these vein, it is taking out. So if you see the cross section, you will find that these blood endothelial cells, and then these are the high endothelial venules, which are the slightly taller cells here. Okay, and now you can see these are the lymphocytes which are coming out, and APCs are coming out. So this is, you know, the structure, and they are coming out from the medulla. Okay, so they are entering into the cortical region, then they are going to the paracortical region, and from paracortical to medulla they are coming out. So this is the general schematic diagram of a lymph node. So the outermost layer, the cortex, contains the lymphocytes, and the cortex has mostly B cells. You know, out of the B and T cells, they mostly have the B cells. Then they have the macrophages, then they have the follicular dendritic cells. Now follicular dendritic cells are different from the normal dendritic cells, which we already seen in the earlier discussion in the last class. Okay, and they are arranged in the follicles. So follicles, we have just seen that follicles are the regions where the B cells develop and B cells are activated. So these follicular dendritic cells, they are the supporting cells, and they form the majority of the follicular region. So that's why these are known as the FDCs, follicular dendritic cells. So here you can see the histological diagram here. So basically, you know, the slide here. So you can see this light shaded. This is the B cell follicle. Okay, this light shaded, and it is surrounded by the dark shade, which is the T cell zone or the paracortex zone. So this is slightly zoomed. Here you can see the follicle. Okay, and then you have the paracortex, and in the follicle you have again a region which is known as a germinal center. Okay, which we will discuss now, germinal center. So under cortex is the paracortex. So we have just seen, so you can see here is the cortex, and under cortex we have the paracortex, and this paracortex is populated largely by the T cells and dendritic cells, because dendritic cells are the specialized cells which present the antigen to T cells. Okay, so they are populated by T cells and dendritic cells, while the cortex is mainly populated with the B cells. Okay, so that's the difference between the cortex and paracortex. Although it doesn't mean that the T cells cannot be found in the cortical region or the B cells cannot be found in the paracortical region. Definitely, you know, you will find the cells there, but it is the majority of the cells are the B cells in cortex and the T cells in paracortex. The innermost layer is the medulla. Okay, we just seen that this is the medulla, and this is the innermost layer of the lymph node. It is the site of lymphocyte exit. So we call it as a egression. Okay, so here you can see the arrow is coming out, and this arrow, when it is coming out, that means the lymphocytes and APCs are coming out. Okay, so this is the site of the lymphocyte exit through the outgoing lymphatics is known as efferent. You have just seen this is the efferent. Okay, so here it is the outgoing efferent lymphatic. So you can see the lymphocytes are and antigen presenting cells are coming out through the medulla, and plasma cells concentrated here. Now this is important, plasma cells. Now plasma cells are what? Plasma cells are the effector cells of B cells. So these are the effector B cells. Okay, B cells, and their major function is what? Their major function is to secrete antibodies. They secrete antibodies. Okay, so their main concentration is in the medulla. So in the medullary region you will find more plasma cells, in the cortical region you will find more B cells, and in the paracortical region you will find more T cells. Now, this antigen, it enters cortex via afferent lymphatic vessels, so that empty into the subcapsular sinus. As I told you that this subcapsular sinus, this is the space. So all the incoming lymphatics, they bring the antigen inside the cortex. And this antigen can be a particulate antigen. That means it may be a bacteria, independent bacteria or virus, or it can be presented with APC. Maybe at the local end, in the local tissue, the antigen presenting cell, it has taken up the antigen and it has processed and presented with the MHC, and that is coming inside using this afferent lymphatic. So antigen can come in both ways, either the particulate or presented with an APC. If particulate, okay, antigen are trapped by resident antigen presenting cells in subcapsular sinus or cortex. So suppose here it is an independent antigen is coming as a particulate. When it will come to the cortical region in this sinus, then immediately it will be taken up by the antigen presenting cells and it will be processed and it will be presented with the MHC molecule. Okay, or it can be presented by resident dendritic cells to T cells in paracortex. Now it may happen that the particle can come and enter the paracortical region. So in the paracortical region we have the dendritic cell concentration more. So these dendritic cells, they can take up, engulf this antigen, and they can present it to the T cells in the paracortical region. If we see the T cells, each naive T cells, it take about 16 to 24 hours to scan all the MHC peptide combinations present by antigen presenting cells in a single lymph node. See how speedily they are working. In a single lymph node, okay, let us see, this is a lymph node, right? And because these are the T cells, so we just discussed that they will be presented in the paracortical region. So let us say it is paracortical region, PC region. In the PC region, if there are antigen presenting cell, now this is an example of an dendritic cell, which is an antigen presenting cell. So all the dendritic cells which are present in a single lymph node, each naive T cell, it can scan all the cells, and this usually takes 16 to 24 hours. So that means within one day, maximum in one day, all the lymph nodes cells will be scanned by each naive T cell just to look for the suitable antigen which can it bind to, and which the antigen which can activate it. So here you can see this is one of the dendrites, and on the dendrites here you can see the MHC molecules which are binding to the TCR. Okay, so if you see here, the pathogen is taken up, and this pathogen is taken up using this vesicle, and then we have the lysosome. So lysosomal enzymes are degrading it, and on the course of the degradation, the MHC molecules are binding the peptide, and then it is presenting on the surface of the dendrite. And when this is present on the surface of the dendrite here, the T cells which are scanning, okay, the MHC peptide combination, then finding the suitable and the specific antigen, they will immediately engage, and this engagement results in the activation of the T cells. And once the T cell is activated, then it will lead to its production of the effector cells by differentiation. All right, so this is how it happens. Now we also have the network of fibroblast reticular cells. So we call them as an FRC. So you can see these FRCs, which are the cells. Okay, fibroblast reticular cells. And these are called fibroblast reticular cell conduit system. It is a conduit system. We call it as a FRCC system, and this guides the T cell movement via associated adhesion molecules and chemokines. So basically this is a reticular cells, that means they form a mesh kind of structure, and they guide the cells. The T cells will glide on it, and then they will send it to the respective destinations. So they have, these are very important because the T cells, they just bind with the FRCC, they are on the network and they glide on these, and then they will keep interacting with whatever in their way the antigen presenting cells are coming. All right, and then if they find the suitable antigen, they immediately engage and then they get activated. So this network is very, very important because it gives a guidance to the T cells. Now, T cells not only associate via adhesion molecules, but also they are released. The chemokines are released, and that also give them the guidance. Because the concentration gradient is built up, and then accordingly our T cells, they find those chemicals gradient and they follow that path. But they are gliding on the this network, this complete reticular cellular network. Antigen presenting cells, they also wrap themselves around the conduits, helping T cells to scan their surfaces. Now, because T cells are coming on these reticular cells, so the antigen presenting cells, they are just wrapping themselves on the surface of these conduits, on the channels, on the pipes, and when the T cells are coming gliding on this, the already wrapped cells which have the MHC molecules and presented on the surface. So these suppose this is the conduit, and here there is a wrap of the antigen presenting cell, and here the lymphocyte is coming, the T cell is coming, it is coming like gliding. When it reaches at the antigen presenting cell which has wrapped on the this FRCC, then they will scan the possible MHC peptide combination, and if the combination is found, then they will immediately engage. If it is not found, they will move on. So that's how it happens. So these APC bound T cells, or the antigen presenting cell bound T cells, they take days to proliferate and differentiate into the effector cells. It takes some time, sometimes days, because they are engaged. When they are engaged, then a lot of signal transduction is happening inside the cell. So we will talk about in the coming discussions, and it takes days to differentiate, and once they are differentiated, they proliferate, they form the effector cells. CD8 positive T cells, they produce the CTLs, the cytotoxic T lymphocytes, to kill, while CD4 positive cells, which are the helper cells, they can differentiate into several different kind of effector cells. So we have seen the few subsets of the T helper cells in the last class. So they can based on the type of antigen and the type of cytokines, they can form the effector cells, while the CD8 positive T cells, which we call it as a T cytotoxic cells, it produces the CTLs, cytotoxic T lymphocytes, which is a, as I told you, it's a kind of the shooter cells, the sniper cells, the encounter specialist cells, which immediately kill the cell of the interest. Unbound T cells exit via the efferent lymphatics in the medulla of the node. Okay, so they have just scanned all the cells, they did not find, then they will be guided towards the medulla and they will exit into the system. Okay, they will not remain within that. So that's how the unbound T cells, they exit from the lymph node. Now focus on the B cells. So B cell activation requires both the antigen BCR interaction, because the BCR is like the same antibody. Okay, we saw in the last class. So this is the antibody molecule, the same surface antibody molecule, which is the BCR. So if the antigen BCR interaction happens here, that is important, and direct contact with the activated CD4 positive cells. So that means it also require the T helper cells. So that is required because we saw that B cells, once they are engaged with the antigen on the be on using their BCR on the surface, then they are partially activated. They are not completely activated unless they move to the region of the T helper cell, the paracortical cell, the region, and there they find, the T helper cells which can give them the complete activation. So that is required. So they are TH cell dependent activity here. So B cells also enter via HEV, so high endothelial vascular cells, and respond to specific signals that draw them to the lymph node follicles. So that is also one of the ways through which the B cells can enter. And they may initially take advantage of the FRCC, just like T cells, because T cells were gliding, on the FRCC. So initially they take the advantage of the FRCC, which guide them, but ultimately they depend upon the follicular dendritic cells. So follicular dendritic cells are their mentors. Okay, so these follicular dendritic cells are used to guide, they are used for the guidance of the B cells. So here you can see these are the follicular dendritic cells, yellow color, and then these are the cells which are on the surface are the B cells. So B cells, they they keep moving on these dendrites here and there, and then from one place they can move to another cell, from another cell they move to third cell, and so on. These follicular dendritic cells, they maintain follicular and germinal center structure and presenting antigen to differentiating B cells. So these FDCs, they have a very important role here. They maintain the follicular and germinal center structures, because ultimately germinal centers, if you will see here, this is the follicle, okay, and this is the germinal center, GC. GCs are the the temporary structures which are formed, which we will see how they are formed. So they maintain this, and they also present the antigen to differentiating B cells, because the antigen is there, so they present it to the B cells. So they do both the works. Number one, they are maintaining the follicular and germinal center structures, and number two, they are also presenting the antigen. Now B cell meets its antigen in the follicle, and then there is an BCR antigen binding. So BCR antigen binding happens here in the follicular region. Okay, partially activated. As I told you that once the BCR antigen binding happens, that only partially activates the B cell. So this partially activated B cells, what it will do, it will engulf the antigen, it will process the antigen and displace it with M.Sc class 2 molecules on the surface, because you know that B cells are also the professional antigen presenting cells. Okay, in the last class, you remember, the B cells are the professional antigen presenting cells. They are professional because they can exhibit class 1 as well as class 2 MHC molecules. So here, once they interact with the antigen, that is activating the B cell partially, and then they are engulfing the antigen, they are processing it, and then they are presenting on their surface with the MHC class 2 molecules. Now they need the T helper cells. So after processing the B cells, they move to T cell rich paracortex to engage possibly pre-activated CD4 positive TH cells, pre-activated, which are already activated cells. That means they are already activated, and they are secreting, they are in the position to secrete the cytokines immediately, because if it is not pre-activated, then it will take some time to activate the TH cells, maybe days to activate the TH cells. But here this is the pre-activated cells. So if, and most likely they will be found here in the T rich paracortical region. So they will go to, and they will immediately upon moving there, the high possibility is that they will find the cells, the pre-activated CD cells, and then they will engage. So they maintain contact for hours because they are already pre-activated, making B cell fully activated and receiving signals that include B cell proliferation. So it is the function of the T helper cell to complete the activation. So the function of the T helper cell is to complete the activation of the B cell. So it completes B cell activation. B cell activation. The second thing is that these cells, they help B cell proliferation, because now the cell is completely activated, so they induce the B cell proliferation. So some activated B cells, they differentiate into plasma cells, because ultimately their effector cells are the plasma cells which lose the BCR. They don't have the BCR on their surface, not the membrane bound form, but the secretory form, because they secrete the antibodies. So some activated B cells, they differentiate into the plasma cell, but other re-enter the follicle, the same follicle, which was there in the cortical region. So but other will re-enter the follicle to establish a germinal center, and this germinal center, it usually is established 4 to 7 days from the activation. So this is so interesting, that the B cells which are moving from the cortical region to paracortical region, there they get complete activation. Some will go to the medulla, because in the medulla you just saw a couple of slides back that there is a concentration of the plasma cells, and these plasma cells are the effector cells. Okay, and here from there they can exit, right? But some cells will go to the cortical region, and in the follicular region they will maintain a center which is the germinal center. So here you can see this is the follicle, and within the follicle there is a germinal center. So a follicle that develops a germinal center is referred to as a secondary follicle. So here you can see in the picture, this is the primary follicle, and in the primary follicle there is no white patch, and this white patch is representing the germinal center. So this is the primary follicle. But as soon as you will see this white patch here, okay, this white patch, this is the germinal center, and when the germinal center comes up in the secondary follicle, when then this in the primary follicle, then it is known as secondary follicle. So you can see the secondary follicle is increasing in size. So not only in the size, but the germinal center is also increasing in size. So here this is smaller, this is small, then this is larger, and here you can see the largest. And then immediately after some time, when the things are over and the immune response is down, then again this germinal center can vanish. So this germinal center, when it comes in the primary follicle, it makes the primary follicle as a secondary follicle. So why do we need these germinal centers? So germinal centers, they facilitate, okay, they facilitate the generation of B cells with greater receptor affinities. Okay, so that more receptor affinities come up, so that more different kind of antigens can be bound. It remains active for 3 or more weeks, right? It remains active for 3 or more weeks. An antigen specific B cell clone, B cell clone means when one receptor, it will bind only to one epitope of an antigen. So an antigen specific B cell clone, it will proliferate and undergo somatic hypermutation. Now somatic hypermutation is something very interesting phenomenon which you will find in the B cells, where the genes are hypermutated. That means the mutation rate is increased by 1,000 to 1,000,000 times more than the normal rate, so that they can generate more different type of receptors. So this is somatic hypermutation, and that happens in the somatic cells, not in the germinal cells. So we call it as a somatic hypermutation. So we will also study this in the subsequent classes, but as of now you should understand that this particular event, which we call it as a somatic hypermutation, it is very important for the antigen receptor diversity, okay, and it will giving more clarity, more specificity, okay. So BCRs with highest antigen binding ability, they survive and differentiate into the plasma cells. Now there is a possibility that there are some B cells, BCR is also binding with the antigen, and other B cells are also binding with the antigen, but the binding is stronger in some B cells but weaker in other B cells. So only those cells which have very strong binding, okay, that will be that will be uh, uh, that will be progressed towards the survival, and they will differentiate into the plasma cells. These plasma cells, they travel to the medulla of the lymph node. So in the lymph node you can see here, this is the medullary region, this is the medulla, and you can see this is the medullary region, and they will just travel to the medullary pathway here. So some will stay and release antibodies into the bloodstream. So they will be here in the medullary region. They will just keep secreting the antibodies, and these antibodies will be flowing with the blood. That is their job. So that's why the medullary region is rich in the plasma cells. So some will stay and release antibodies into the bloodstream, while others will exit through the efferent lymphatics. Okay, they will exit and they stay in the bone marrow, where they will continue to release antibodies into the circulation. Okay, so their primary job is to secrete the antibodies. Some will stay in the medullary part of the lymph node, others will go to the bone marrow, and then there they will keep secreting the antibodies. T or B memory cells, T or B memory cells, they can stay in secondary lymphoid tissues or can exit the nodes and travel to among tissues that first encountered the pathogen. So that means the memory cells, either they can stay in the secondary lymphoid tissues or they can go at the site of the infection from where the antigen was brought into the local lymph node, right? So that both ways they can happen. So memory T cells, now we are talking about the memory T cells, and you remember these memory cells are important because they are important for the secondary response, right? So memory T cells that reside in secondary lymphoid organs, okay, when they choose to reside in secondary lymphoid organs, they are referred to as central memory T cells. So those cells are known as central memory T cells, which remain in the secondary lymph node or lymphoid organs, and they are distinct in phenotype and functional potential from effector memory cells. Now another kind of memory cells are the effector memory T cells, which circulate among the tissues. They go from one tissue to another, and they go into the tissues and they remain there, while some remain only in the lymph node. So these are the central, while these are the effector, because they are going at the site of the infection from tissue to tissue, and then they are looking for their respective antigens. Okay, the third population is the tissue resident memory cells, because some residents when they will, sorry, the some T cells, effector T cells, when they will circulate among the tissues, they remain resident in the tissue. These are found in the peripheral tissues for the long term, and these are the first cells respond during the secondary response. So these are the first responders during the secondary response, and these memory cells are known as tissue resident memory cells. Now, after the lymph node, spleen, which is the largest secondary lymphoid organ. Now you can see here, this is the spleen, and it is located in the abdominal cavity. So you can find the stomach or abdomen here, and then you have the spleen, and this spleen has a very rich blood vascular supply. Okay, so we have the artery, which is the red one, we have the vein, we have the capsule, because this is also capsulated. Then this capsule is invasing and it is forming trabecula, and then we have the vascular sinusoid, which is again this space, and then we have two distinct regions. We have the white pulp, okay, these lighter regions, and then we have the red pulp, which are the red pinkish region. You can see it here. So this is the general structure of the spleen. So spleen, now it is important, and it's a very important organ. Is spleen specializes in trapping and responding to blood-borne antigens, not tissue-borne, but blood-borne. While our lymph nodes were specialized, or they are specialized for tissue-borne infection. So spleen is for blood-borne antigens. Systemic infection. Why we call it systemic? It is not localized, because blood vascular system, it flows through each and every tissue. And once the infection is inside the blood, that means the infection is systemic. Now it is in the complete system, right? So we call it as a systemic infection. In case of tissue, the infection may be localized. It may be just present in a particular tissue. For example, it may be present in the just kidneys, or it may be present in the liver, or it may be just present in the stomach, or it may be present in the throat, or it may be present in the lung. It may be localized, not systemic. But when it is present in the blood vascular system, then it becomes a systemic infection. And unlike lymph nodes, okay, they respond to antigen which are drained from local tissue, because lymph nodes are drained from local tissue, while the spleen, it has the antigen from the blood flow. Spleen is not supplied by lymphatic vessels. Now it is important, and a very important difference. Not supplied by lymphatic vessels. But we have the splenic artery. So you can see this artery, splenic artery, which takes the antigen in. And then we have the splenic vein, which takes it out. So you should understand that spleen is supplied by splenic artery, and then the exit of the antigen is by splenic vein. The number of lymphocytes which pass daily through the spleen is greater than all the number of lymphocytes which are present in the lymph nodes combined. Whatever the number of lymph nodes we have, although these are small structures, but they are spread it. Whatever the number they have. So on the daily basis, if we see the number of the lymphocytes which pass through the spleen, it is much more than the total number of the lymphocytes which pass through all the lymph nodes together. So you can see, how important this organ is. The capsule, which is here, it extends into the interior and dividing into lobes. So you can see the lobes, and all the lobes have the same or similar function. Each splenic lobe has red pulp and white pulp. So you can see here we have the red pulp and we have the white pulp, and it is separated by a specialized region, and which is known as the marginal zone. This region is known as the marginal zone. So here we have this, the marginal zone. So marginal zone is basically the region which makes a boundary between the white pulp and the red pulp. Red pulp has RBCs, macrophages, and some lymphocytes. This red pulp is the graveyard of old RBCs. This red pulp is important for iron metabolism by macrophages. We have special macrophages for iron metabolism. It is also the site for platelet storage and also the site of hematopoiesis. Okay, so red pulp is very important. It is the graveyard of old RBCs. It is the site of iron metabolism. It is the site of platelet storage, and then it is also the site of the hematopoiesis. While the white pulp, it has B cell follicles and the periarteriolar lymphoid sheath, which we call it as a PELS. And this region is populated by T lymphocytes. So we have white pulp, because this is white, so you can just for the sake of remembering, white blood cells, rich region is the white pulp. And since white blood cells are the B cells and T cells, so B cells are in the follicles and T cells are in the PELS. Then germinal centers are generated within these follicles during an immune response, because germinal centers are important for the temporary effectiveness of the immune response. So they are generated within these structures also. The marginal zone, because it is a boundary between the red pulp and white pulp, it is a specialized cellular region between the blood and the white pulp, because red pulp is rich in blood supply, because you need to have the RBCs there for dumping. So blood supply is there, so that's why it is red, because it has a more vascular supply. So it is the specialized cellular region which separates the red pulp from the white pulp, and it is populated by specialized dendritic cells. Though these dendritic cells are special dendritic cells. Then we have the specialized macrophages, and we have a unique B cells. These B cells are different from the normal B cells, and we call them as a marginal zone B cells. We call them as a marginal zone B cells, or in short we write MZB cells. So these MZB cells, they are slightly different from the regular B cells. So let us see how these are different. So these marginal zone B cells, they are strategically located between the circulation and the white pulp, providing the first line of defense, right? Providing the first line of defense. So in the picture you can see this is the marginal zone, all right? And on the marginal zone you will find this is the B cells. So here you can find the B cells. This sky blue color, these cells are the marginal zone B cells. While in the follicle we have the regular B cells. This purple color are the regular B cells. Okay, this is the regular B cells. So these are different. They are here standing as a first line of defense. So you can see here. So this, they are forming a first line of defense here. Okay, they are forming a first line of defense here. They express, so these marginal zone B cells, they express both innate immune receptors. Okay, for example, TLRs. TLRs are, remember, toll-like receptors. So they express both innate immune receptors, for example TLRs, and unique B cell receptors, okay, that recognize conserved PAMs. Now what are PAMs? Tell me. Okay, pathogen associated molecular patterns, PAMs. Remember from the first class, PAMs and these TLRs. Okay, so these are the innate immune receptors. So they are expressing innate immune receptors as well as the BCRs. Now this makes them different from the regular B cells. Upon binding antigen, okay, because they will bind the antigen to their BCRs, cells rapidly differentiate and secrete antibodies. Right? You remember the regular B cells, they need the help of the TH cells, and TH cells will give the complete activation, then only they can be differentiated and forming the effector plasma cells. But here, upon binding, independently to T cell binding, they can secrete the antibodies. So that is the major difference between the splenic marginal zone B cells and the regular B cells. So therefore they can be activated either in T cell dependent or T cell independent manner. Okay, now here comes the difference. Remember this. That is the differential feature between the regular B cell and the splenic marginal zone B cell. Now let us just see some more differences between this regular or follicular B cell and MZ B cells. So their primary localization, they are in the secondary lymphoid organs, mainly in the B cell follicles, while the MZ B cells, they are also in the secondary lymphoid organs but in the splenic marginal zone. That's why they are known as the marginal zone. Then they have some distinguishing surface markers. Now you can see here surface markers are almost the same, but you can see here low and high. So low, that means low, and high means high, more expression. So one thing which I want to highlight here is IgD. So IgD expression is high in case of follicular B cells, while in marginal zone B cells this expression is low. Although it is there, expression is there, but the expression is low in case of MZ B cells and expression is high in case of follicular B cells. Apart from that, you can just see it is 220, this CD19, here it is low, positive, it is high. Then here CD1D is low, here it is high. It is CD21, this is high. It is CD23 high, this is CD23 low. So these are some of the marker differences, and that makes easy for us using flow cytometry to isolate these cells. Now principal function we have just seen, they participate in T dependent immune responses, follicular B cells, right? While in case of MZ B cells, they have the housekeeping function. So they provide natural IgMs, okay, which are present in our blood, and they are the first responders to blood-borne pathogens because they make the first line of defense. Marginal zone, which lies the boundary between the red pulp and white pulp. Okay, I hope by now you must have understood the difference between the marginal zone B cells and the regular B cells. Okay, so now this is a very general, diagram which shows that, how it works. So we have the antigen, and this antigen, it is entering the marginal zone through splenic artery. So splenic artery is the main supply. Our B lymphocytes and T lymphocytes are also coming through splenic artery, and they are reaching to marginal zone. So the first interaction between the antigen and our lymphocytes that takes place in the marginal zone. So here the antigen is trapped. Marginal zone B cells, they bind via complement receptors because they have the innate receptors, and they are also processed by DCs. Okay, and once the marginal zone B cell binds via complement receptor, they convey it to the follicular or the follicles in the white pulp, right? So they took the antigen to the white pulp so that the regular B cells can now take over. Now because they are also processed by the dendritic cells, and you know that dendritic cells, they report to T cells, so they will move to pels, which is the white pulp, and then they will interact with the T cells, right? So that's how it happens. So now this is the same picture, same diagram. Okay, so here you can see the green part, which is the T cell zone, and here you can see the T cells lined up. Okay, so these T cells are different from the B cells which are in the follicles, and these MCB cells which are in the marginal zone. So naive B cells which are unexposed to antigen, they interact antigen in the follicles, and naive T helper and TC cells meet antigen presented by DCs in the T cell zone, which is the PELS, the green zone. Once activated, the CD4 positive T helper cells, they provide help to B cells and CD8 positive T cells that have also encountered antigen. So the main function of the T helper cell is to give help to both B cells and the CD8 positive T cells. Some activated B cells together with some T helper cells, they migrate back into follicles and generate germinal centers, because here also they have to generate the germinal centers. So germinal centers are nothing but it is a sort of, the temporary structures which are made during the immune response to heighten and to, optimize the response. Now, after the lymph nodes and spleen, we are moving our discussion towards the barrier tissues, and these barrier tissues have MALTs. MALTs are the mucosa associated lymphoid tissues. So tissues like skin, okay, mucosal membranes of the digestive, respiratory, and urogenital tracts, they also have the T cell zones and lymphoid follicles. So lymphoid follicles are for the B cells, okay, and we have the T cells zones. And epithelial cells in these organs, in case of skin, there are multiple layers of epithelial cells, and in case of mucosa, we have single layer of epithelial cells. So that's the difference between the skin and the mucosa, and both need to be protected. Why? Because if you see the combined area, it is around 400 square meter. So that is the size of the football stadium. So that much area is there if we both combine of the skin and mucosa in an average adult. So now you can understand that we need to protect it. Otherwise, the antigens will immediately breach and they will start destructing our tissues. So these surfaces have organized lymphoid tissues, and we call them as a mucosa associated lymphoid tissues or MALT. Okay. Now, sometimes these MALT have some specific names based on the location. Bronchus associated lymphoid tissues. So if they are present in the bronchi of the lungs, we call them as a bronchus associated lymphoid tissues or BALT. Okay. Nasal associated lymphoid tissues, if they are present in our nasal cavity, in the nose, we call them as a NALT. Then we have gut associated lymphoid tissue if they are present in our alimentary canal. And then if they are present in the skin, we called them as a skin associated lymphoid tissue or salt. So balt, nalt, galt and salt, these are some of the tissue specific malts, but they are malts. Okay. Now, if we see the galt, we have many types of gut associated lymphoid tissue. Gut associated lymphoid tissue. These are the well organized structures. We call them, or we know by the names of tonsils and adenoids. And these tonsils and adenoids, they make the Waldear's tonsil ring. Now you can see here we have the Waldear's tonsil ring. So you have seen this is the adenoid. Then we have the tubal tonsils on both sides. Then we have the palatine tonsils, or what we call them as a tonsil per se. It is the palatine tonsil and it's a kind of tonsils. So it is also on the both sides. And then on our tongue we have the lingual tonsil. So when we see, they form a ring-like structure, and this ring of tonsil is known as the Waldear's tonsil ring. Loose, barely organized cluster of lymphoid cells, because they are just cluster of lymphoid cells and they're loose, barely organized. They are not as organized as we have just seen in case of spleen and lymph nodes, but they are barely organized. Now, lymphoid cells are found in various regions within the lining of the intestine as well, because we are talking about the GALT, which is the gut associated lymphoid tissue. So after mouth, we also have in the intestine. The outer mucosal epithelial layer contains intraepithelial lymphocytes, and many of them are T cells. So if you see the mucosal epithelial layer, it is the mucosal epithelial layer. This layer is the mucosal epithelial layer. And here you will find intraepithelial lymphocytes. That means the name itself is saying lymphocytes which are present inside the epithelium. So we call it as a intraepithelial lymphocytes. And many of the lymphocytes are the T cells. Then we have the lamina propria. Lamina propria is what? It is the tissue, okay, beneath the epithelial cells. So here you will find the lamina propria. This is lamina propria. They have B cells, they have plasma cells, and they have activated T cells and macrophages in loose clusters. So you can see here, there is a clustering which is here, and they are very loose. That means they are not organized. So you will find some areas highly concentrated of a particular cell, some areas are mixed. So they have a loose clusters. More than 15,000 lymphoid follicles within lamina propria of a healthy child. So you can see, because children usually have very high response, immune response. They are undergoing because they are in the formation of making memory cells. Okay. So more than 15,000 lymphoid follicles are there within the lamina propria of a healthy child. And we have the Peyer's patches, which are the nodules of 30 to 40 lymphoid follicles. So you can see here, this is the Peyer's patches. Okay. And this Peyer patch, it has around 30 to 40 lymphoid follicles we can have, and it can extend into the muscle layer just below the lamina propria. So this is the muscle layer. You can see the muscle layer here, and this can extend towards it, right? So you can see, this is how they are structured. Lymphoid follicles can develop into secondary follicles with germinal centers. So you have just seen that when a primary lymphoid follicle, it develops the germinal center, then we call it as a secondary follicle, and you can find this secondary follicle here. So the number of plasma cells in malt exceeds the sum total of plasma cells in the spleen, lymph nodes and bone marrow. It's very, very important to know. You can see, in the entire gut associated, uh, tissue, the sum total of the plasma cells which you are finding, if you number them, then the entire number is much more than the number if you combine in the spleen, lymph nodes and the bone marrow. So how active it is? Why? Because it's a very long organ. Okay, and then at each and every point you need to have these effector cells in order to protect our gut. So we need more number of cells, and the cells are exceed just like anything, enormous number. Okay. The epithelial cells of mucous membrane, they deliver, now important, they deliver small samples of antigens from the respiratory, digestive and urogenital tracts to the underlying malt. Because we have the mucous membranes in the respiratory system, in the digestive system and in the urogenital system. So what they do, when they find the antigen, they deliver it to the underlying malt, and malt is always below the epithelium, right? Now we have some specialized M cells. So these M cells are the special cells which transport antigen across the epithelium in the digestive tract. So here you can find, this is the M cell, this is structure. So you know this is sorry this is the M cell here. This is all M cell. Yeah, this is M cell. And these are the specialized cells. These are flattened epithelial cells lacking the microvilli. So you can see the neighboring cells. So neighboring cells have the microvilli. These are the microvilli. Okay. Here also we have the microvilli, right? But these cells, M cells, they are lacking. They don't have. They are the flattened cells, relatively flattened cells. And they have the deep pocket in the basolateral membrane. So this is the basolateral membrane. Okay, we called it as a BM membrane, BL membrane, basolateral membrane, and they have the deep pocket. Now this is the pocket. Okay, you can see here, this is the whole deep pocket. It is filled with B cells. So you can find the B cells here. Okay, T cells. This is the blue color is the T cell here. Okay, we have the B cell, we have the T cell, and macrophages. So here it is the macrophage. Okay, understood? What is the structure of the M cell? It is relatively flattened cell, no villi, microvilli, and they have a pocket. And this deep pocket is towards the basolateral side. And this is filled with the B cell, T cell, and the macrophage. And the function is to transport the sample of the antigen. So this antigen, it will be transported as a sample, just to show that what kind of antigen it is. Okay. So antigens in the intestinal lumen, they are endocytosed to the underlying pocket membrane, delivered to lymphocytes and antigen presenting cells, and most important to the dendritic cells. So here you can see, this is the antigen which is endocytosed, and then it is giving and delivered to the various cells. This leads to the activation of the B cells that differentiate and then secrete the IgA. IgA is a type of antibody, class of antibody, which we will see in the next class. So this IgA, by now, I mean, just for your information, this IgA, it is a kind of 5 type of antibodies which we see. So this is 1 out of 5. So IgA is a special antibody, and it is secreted here in this region, and it is concentrated in the milk. Okay, and is used to fight various type of infection at the mucosal site. So this is important for the mucosal immunity. IgA is important for mucosal immunity. Immune cells, which are the macrophages, the dendritic cells, intraepithelial lymphocytes, they sample antigens from the lumen, and with Treg cells, remember Treg cells, the regulatory T cells, they work to distinguish between the beneficial commensal bacteria and more dangerous pathogens. Now, it is important to note here that our gut has some good bacteria, and we need them. They are the commensal bacteria. We need them because they help in our digestion process, right? So, and sometimes what happens, because of some wrong food or our poor hygiene, we introduce the germs in our gut. So when they mix with the commensal or the commensal bacteria, then they are sampled along with the commensal bacteria. And because commensal bacteria are the part of our gut system, so they will be our immune system can easily differentiate that this antigen is ours and this antigen is a new antigen, which is a foreign antigen. So they will activate against the foreign antigen. So that's how it is important, and they can distinguish what is, between the beneficial commensal bacteria and the dangerous pathogens. This help to maintain tolerance to commensal microbes that coexist at our surface. So that gives the answer, right? Now, skin. Skin is the largest organ in the body, and it is the part of the innate, that is non-specific immunity. Just, if you see the structure, we have the epidermis and the dermis, and epidermis has keratinocytes, okay, which makes our skin impermeable to water. Then they have some Langerhans cells, which are, phagocytic cells. And then we have, in the dermis, we have the blood vessels, we have the dendritic cells, we have the lymphatic vessels. That's how it is organized. So keratinocytes, they secrete a number of cytokines to induce a local inflammation. So whenever there is a insect bite, or whenever, like, some rubbing, rashing, or some antigen is there, then our keratinocytes, they secrete cytokines which induce a local inflammation. So inflammatory cytokines are secreted. Then we have the Langerhans cells, which are skin resident dendritic cells, which is, they have the phagocytic activity, and they are scattered among the epithelial cell matrix of the epidermis. Okay, so in the epidermis you can find these Langerhans cells. They undergo maturation and they migrate from epidermis to regional lymph nodes. So after maturation, they just migrate, where they activate the naive T cells, because if they have antigen, they will get differentiated, and then they will activate the naive T cells. Dermis also contains scattered immune cells and hematopoietic stem cells. So our dermis also have hematopoietic stem cells, right? So you can find the hematopoietic stem cells here also, not only in the bone marrow, but here also, that comes from the circulation. Most skin lymphocytes are either previously activated cells or the memory cells. Why? Because they immediately need to recognize. Our skin is at the border between the external environment and the internal environment. So we need to, put our best guards there, so they can immediately recognize that there is an infection, and without losing any time, they just act upon it. So that's why they have the either activated cells or the memory cells, and memory cells are generated because of the previous infection. All right. Now we also have the tertiary lymphoid tissue. So tertiary lymphoid tissues are the sites of active infection and immune activity. Now, apart from the primary lymphoid tissue and the secondary lymphoid tissue, now if there is some infection anywhere inside the tissue, in the body, in any organ, any place, then it can become a tertiary lymphoid tissue. How? Activated lymphocytes from the secondary lymphoid organs, they can return to the infective tissue. Now you remember that this infected tissue can be any part of the body. Okay, any part of the body, any part of the body, as effector cells, because these lymphocytes, they are activated from the infection, and then they have generated the germinal centers in the SLOs, and then they have returned, the effector cells are returning at the site of the infection, and they can reside there as a memory cells. Okay, so they can reside there as a memory cells. They create microenvironment for the returning lymphoid cells. So tertiary lymphoid tissues also make, a microenvironment for the returning lymphoid cells. Now how it happens, you can see here, in the first stage there is a mixed T and B lymphocytes. There is no follicular dendritic cell and there is no germinal center. So you can see this is just a loose cells, okay, at the site of the infection. In the second stage, B cells, they cluster and they are surrounded by this T cells, all right, and then they are have follicular dendritic cells, but there is no germinal centers. So you can see here the follicular dendritic cells, but we don't have the germinal centers. But in the third stage, B cell cluster surrounded by T cells, we have the germinal centers, and then we have the presence of the HEV and LEV. So that means we have established a secondary tissue, a secondary lymphoid tissue kind of structure at the infectious site. But this is not a regular tissue. It was established against an infection or in response to the infection. That's why we call it as a tertiary lymphoid tissue. We call it as a tertiary lymphoid tissue. So this is established and it acts like a secondary lymphoid tissue. So we have the T cells, the blue color, we have the B cells, the yellow color, and then we have the germinal centers. You can see the darker area, the darker patch, just like in any secondary lymphoid tissue. Okay, so now we will just see that we have the distribution of the lymphoid tissue all over the vertebrates, but what kind of lymphoid tissue you will find. So here we have started with the lower vertebrates and here we go to the higher vertebrates. So if we see here in the lower vertebrates, galt is present in all the vertebrates. Okay, this is the tissue which you will find in all the vertebrates. Then more specialized thymus slightly and spleen, they have started coming from trout. Okay, and then from frog you will find bone marrow. From chicken or birds you will find the lymph nodes and the germinal centers, and from mouse and then humans here, right? So galt is the primary tissue which is present everywhere. Primary matlab not the primary, but primary is like the first tissue, which is the first secondary tissue, I would say, which is present in almost all the vertebrates. But thymus, spleen, bone marrow, lymph node and germinal centers, they are coming up slowly, and they have developed through various processes of evolution, and we have basically specialized them. So vertebrates, they are getting more and more complex in terms of our immune system, starting with the galt, then they are developing thymus and spleen, and then they are bone marrow, lymph node and germinal centers. So that's how the evolutionary distribution of lymphoid tissue is among the vertebrates. Fine. Okay, so now we are closing the discussion for the class today, and then let us summarize what we have discussed. So we have discussed the primary organs, which are the bone marrow and thymus. We have discussed the secondary organs, lymph nodes, spleen and tissue like malt. We also discussed the tertiary lymphoid tissue organization and how it is made and how it is established, and then some evolutionary distribution. I hope you have enriched some knowledge about immune system, and you are exciting and you are liking this. In case if you have any comment or if you want to share your feedback, you can, my email is given here, you can reach out to me.